Yeah, thank you for this opportunity to share our recent paper on Bayesian safety surveillance with adaptive bias correction. So this is actually a collaborative project with the OHDSI Methods Work Group and the US FDA CBER Center. The task that we received is to help the FDA to improve their procedure for vaccine safety surveillance. The overarching goal of safety surveillance here is to detect if a vaccine product increases the risk of certain adverse event of interest. Now, in pre-market clinical trials, the sample sizes would be too limited to detect some rare but severe adverse events. That is why we have to rely on post-market surveillance once the vaccine has been approved and has been on the market. Statistically, this is a hypothesis testing problem. We want to test using the data to see if the vaccine does indeed increase the risk of some adverse event. Now, because we want to be able to rapidly detect any safety signals of concern, we have to rely on sequential analysis of data in the real world as they accrue over time, usually in monthly frequencies or sometimes bi-weekly frequencies. So generally speaking, the framework that we are developing here are not just limited to vaccine safety events. It can be generalized to any kinds of surveillance problems using streaming data. Now, going back to the problem at hand, this is actually quite challenging because we have to sequentially look at the data and come up with some test statistic and compare it with some kind of decision threshold. At the time point where the decision threshold is crossed, then we stop the study and declare some kind of safety signal. But the problem here is that as we repeatedly analyze data over time, the type 1 error rate or the false positive rate can be blown up. This is called sequential multiplicity. Now, there is a widely used standard approach called max SPRT that can theoretically address this issue and is also adopted by, for example, FDA and the CDC. But the problem here is that in order to pre-specify this decision threshold to maintain, say, an alpha level 5%, it has to rely on a pre-specified analysis schedule. And we have to stay committed to this pre-specified analysis plan. There's no way to adapt it if things change in reality. So it's very inconvenient to use as a framework. On top of that, we have to work with observational data instead of experimental data, so including insurance claims and EHRs and so on. And as we know, within OHDSI, there is intrinsically systematic error with those data sources that can lead to bias in estimation and inference. And in this context, this can hugely inflate testing errors, leading to unreliable decisions. And in this rather rigid framework, there's no statistically coherent solution for addressing and correcting the bias. And actually, in our empirical evaluation, we have found that if you implement this max SPRT approach with a pre-specified type 1 error rate level, say alpha 5%, then what you truly get at the end in terms of the empirical type 1 error rate can be as high as 27%, so more than 5 times higher than your assumed alpha level, meaning that you would be detecting way more vaccine safety events than reality. So you're telling the public that the vaccine is not safe when the vaccine is actually quite safe. So that is very concerning, very unreliable. And because of those reasons, we have been working with the FDA CBER Center to try to develop a better solution. We want a more flexible framework so that we don't need to rely on a pre-specified schedule for years into a vaccine campaign. And we want to reduce on the bias so that we can produce more reliable results. So in order to have a more flexible framework, we propose in our paper to use a Bayesian sequential analysis framework instead. So instead of looking at p-values over time, we now instead look at something called the posterior probability of the hypothesis, conditional on the data over time. So from a Bayesian framework, this is more naturally a sequential quantity that we can easily compute and update over time. And more importantly, we now no longer need to rely on a pre-specified schedule. Everything is now more adaptive given the data evidence accumulated over time. And this quantity actually quantifies how much data evidence we have collected is in support of the statement that the vaccine does elevate the risk of the adverse events. So it's more interpretable than a p-value as well. And to address bias, we actually use a very commonly used tool for OHDSI network studies. We use negative controls. By definition, a negative control outcome is an outcome that we know is not associated with the exposure of interest. Now, by analyzing a large set of negative control outcomes, what we can get is an empirical estimation of the distribution of the bias in our analysis framework and in our data source. Through a Bayesian joint model for all the negative controls and the outcome of interest, we can jointly get an estimate for the effect size of interest that is bias corrected. And more importantly, we can do this adaptively over time in this sequential analysis framework that presents a very coherent statistical solution that preserves all the nice properties. In our paper, we have done a large set of empirical evaluation experiments to compare the properties of our method against the standard method MaxSPRT. So going back to the type 1 error rate chart that I showed you previously, in this example, our Bayesian adaptive bias correction method is able to bring down the empirical type 1 error rate to a level that is very close to the nominal desired level that which is everyone's favorite, alpha equals 5%. And on the other hand, we actually offer more statistical power in detecting a true positive signal. So if the same amount of type 1 error rate is allowed between the standard approach and our approach, we're actually able to offer much more power in figuring out if some signal is an actual safety signal. And that is particularly the case for a smaller effect size. And those effect sizes are very close to the actual effect sizes for vaccine safety events in real life as well. So this means our approach, being a better solution, can bring down decision errors while providing more power and faster detection of true safety signals. Now, this is actually my last slide, but I want to conclude and emphasize that this is actually a team effort with a lot of collaborators and data partners within OHDSI and with collaboration with regulatory agencies like the US FDA. If you're interested, please check out our R-Shiny app for all the evidence that was produced in our paper. We have also implemented the method in the evidence synthesis R package, which is part of the Hades software suite as well. And here's a barcode for our paper. Thank you.